Hello students, welcome to EPG Pathshala. I am Indira Ghosh from JNU and today we are going to talk on module application of designing using bio and chemo both informatics and under the paper computational biology. Our learning objective is we should learn rational thinking that you have the drug design and biology together and we will apply it to infectious disease. And my examples will be anti-malarial and anti-tuberculosis. To revise your concept of ideas, what I have talked earlier modules are protein structure starts from sequence. However, it has some prevalent sequence from sequence to a secondary structures, which is shown here as the alpha helix, and then you get a tertiary structure, that is combination of those alpha helix or beta structures, and come up with a proper functional protein. But these folds or these proteins domains would contain one of these very specific motifs called the active sites. This may be disjointed sequences and structurally, or they may be consecutive places as well. Here I show you two tertiary structures. One is called myoglobin, which is a part of a hemoglobin, which is the major portion of protein available in our human blood. Also, on the right hand side, I have shown you another protein called concanavalinae, which occurs many a times and very prevalently in plant cells. Now, two structures are very predominantly can be found out. One is left side shows the structure of alpha helix, and right side shows you the structures contains only in beta. Not all protein have only alpha or only beta. There could be proteins which has alpha, beta, beta, alpha, many combination of structures. But most importantly, we should go, which will be shown in next slide, that is how we represent these structures. Here is the first picture shows you on the left hand side, one small molecule as well as the protein can be shown as a line diagram, then ball and stick model, and then you can have a space filling model, that is which it shows the whole structure. However, how to build a molecule is shown in the bottom. There would be not only the bond length and bond angle of this, every atom are connected in a protein or in a small molecules, also their orientation, or if you consider any three atom in the bottom picture, the three atom containing plane and another three atom containing plane would be possibly rotate around them by the bond shown in middle, that is second and third, those two atoms, then you can have different orientation possible. Now, if you consider my thumbs to be associated with the last or extreme two atoms, then the orientation of these two atoms can change just because these two planes are changing. This concept was actually ported, and next slide will describe that concept, which was done by Linus Pauling very early time, and later on whole map developed by J.N. Ramachandran. I would like to show you the Indian legacy in the field of protein structure modeling topics. Linus Pauling actually found out quite long back in 60s that the alpha helix can be stable, one of the secondary structure as shown previously, and they occur in protein quite a plenty, and they are stable only by the way that they have an intrahydrogen bond, that is along the chain they have intrahydrogen bond, and this goes very nicely repetitive. Professor J.N. Ramachandran in India approached the same problem in a different way. So he took and developed a contact map by calculating every peptide or every protein's build, that is the smallest unit you require to make it to work as the rotational angle, which he defined and which decides the orientation between two atoms. I had in my earlier topics, I have gone through this in detail. Here I will just show you that this plot, which is called Phi-Psi plot or called the contact map plot of proteins, has been identified in 1967-68 and published those days. Very nice concept. Till today, all the protein structures came out, seems to be behaving exactly according to these particular plots. This contribution has led us to model a lot of molecules or lot of protein structures which we could not have done if the crystallization is not available. Many crystals may not be available as a crystal structure, many proteins. However, homology modeling, which I will talk later, you will see has been highly beneficial to use these plots. So not only the crystal structures, but even the new structures comes out, we know they will be falling in this allowed criteria or allowed region, which are shown in dark black. This is the theme topics for today's talk. Now, how one finds medicine. Actually, how it used to be before the biology or genomics of plenty of genome systems came out, that is not only pathogen as well as several species and human genomes, etc. It used to be driven by chemistry. So major work is to be done by designing a chemical in the early phase, which will be look like substrate or modified substrate or some such things. Then understand how they react to the species or proteins or receptors, etc. And then they used to take these examples and try to treat in vivo to find how it is associated to the disease. This particular direction has been totally changed due to the impact of omics, that is the genomics, proteomics, etc. So now available information are churned out in a very different way. So the future generation from 2000 onwards, most of the pharmacological data involvement as well as biological data involvements are done in a very early phase. And so understanding of disease and their informations are collected first. Then understand how the biology of this disease to be tackled with, what could be the sufficient target or receptor, etc., which should be modulated or inhibited fully to get the required involvement, required survivor or from disease. Then one uses leveraging that information, using that information, design a chemistry. So as you notice, because of the impact of this omics has changed the direction totally. The difficulty or challenges in drug development. There are noted here a few facts which you can read it by yourself. Only it tells that more and more the days are coming up, we are hardly able to find a novel drug. Now, why it is so? Number one reason is, however we know a lot of genomes, we had become almost saturated to find targets. I addressed this issue in my previous module, and which addresses the issue of new targets. Let us go to the next. How good or bad it is for modeling if you want to model the chemical side of it. This module's objective is to find the relation between biological and chemical by using all kind of computational biology techniques. Noticeably, you must see this, that not at all except one new drug has come out last 50 years for tuberculosis, malaria, MRSA, etc. So that shows you how bad situation we are in regarding pharmaceutical companies, etc. We now talk about three different topics basically, and the first topics is what we would like to emphasize, and which I'm going to emphasize in my next few talks, and that is structure modeling. That is how we model a structure, and interaction mapping. This will lead to some of this drug design. So there would be another topics, which is chemoinformatics and QSAR driven designing of molecules. That will be in my later part of this module. So what is interaction and drug design, the relation between these? Basically, first we need to know the structure and display the structure first, so that we have a real visual idea about the interaction between domain. On the left hand side, I have shown you one of the protein, and it is a homodimer protein. It is only active, its ICDA structure, which is homodimer is active. So in case of interaction mapping, we have to keep in mind, if it is a homodimer, we have to find the active sites. Sometimes homodimer might have two interaction sites or maybe single active site as well. Second picture shows how nicely we can see the active site. You can see the active site as if it is a groove, one small groove where something is colored in little buff color. Now, if we design the molecule to fit in, we need even more information than only showing you 3 dimension, which is shown in the next. The interaction distance between each of the amino acid and the molecule where will be designed there. The bottom picture actually shows the same thing, but when it is membrane protein. So as we gathered, we will go ahead. I will try to show you a case study of a soluble protein which is on the upper. But nonetheless, the same system can be simulated and designed and modeled and take the work done also as a membrane protein. I would like to discuss here one of the important designing, and our objective is here screening compound has been sent by high throughput screening, and we want to design a compound from started from this screened compound chemical natural product library. But our objective is to find a selective inhibitor towards the plasmodium, and this selective inhibitor should inhibit, because the biochemistry is done on plasmepsin 2, which is an acid protease, and has been already known very well that this is a anti-malarial. So you inhibit a plasmepsin 2 or all the plasmepsins, you get very high quality, good anti-malarial activity. So this is a case study I taken to show you how actually you apply the techniques which I have gone through with you in the whole module. Here we use molecular modeling. We use all these pictures and identification of active site, etc. shown here in colors. You can see the 3 colors are depicted. One is yellow, is the all the active site is been colored. Yellow color is neutral, and red and green are acidic and basic. But you should be noticed there are smaller active sites also has been depicted or found out. However, the main substrate or inhibitor lies in the large pocket. Hence, when we will do the screening or we will dock or we will design compound, we will use the major active site. Queensland Pharmaceutical Research Institute had a huge amount of natural product library. They scanned this compound, and I was working with AstraZeneca at that time, and we identified these compounds called ARQ104. What we figure out that plasmodium inhibition, it is 20 micromolar, and enzyme inhibition, it is 10 micromolar. So the compound is not very bad or very poor. However, what we notice in the beginning, selectivity, that is, even it inhibits the cathepsin and pepsin, which is human enzymes equivalent to plasmodium, is almost the same amount. Our objective was design the selectivity, and that is highly important because plasmodium is having a host as human. So we require to increase the selectivity difference. I will show stepwise many work of which are already depicted and discussed in previous menus. Basically, what we approached was to do a structure-based drug design. What I did actually is map the active site properly, as I've shown you, then design the interaction possible. Map the active site is shown here, and you can see the 2 aspartic system or 2 aspartic diodes in red blobs in there, and we showed all the amino acid which are interactive over there. The publication is cited on the left. Then we mapped active site and then put or identified by using grid another set of program to identify what would be the interactive sites which are still available. Using another software called Ludi, which actually new version of these softwares are available on BioVia now, also available for academics, freely downloadable, in which is called GrowMol, the software package. But what it does is it takes the interactive sites, puts the molecule which we already got by screening, and design different kind of small fragment based designing, and then identifies which can have the best interaction. In the bottom right, you see it has been got not to achieve this golden color molecule is the fragment. If we add up to this fragment identified compound, that we can achieve a very good selectivity. How we know that? This is the results. That is, we did design the compound as it's shown. So bottom compound structure is shown as QPRI or AIRQ104, which was converted into another fragment A designing, where we have used one small left hand side. You can see a small molecule, small fragment that achieved hydrogen bond and snug fit in the available pocket. Top picture actually shows you how many trial we have done. Many of those small mesh like structure on the Ludi shows us that it is up there, that many of them has been tried. So we got the selectivity, and the selectivity was 10-fold more. We not only lower the enzyme activity with acid protease or PLF, and we figure out that this particular hydrogen bond cannot be formed in the human corresponding acid proteases and doesn't even have space enough to snug fit. Hence, the increase of selectivity was 10-fold. So this molecule was taken for next level of designing for other experimental and quantitatively find the solubility, etc. Another experiment, what I would like to say, and that this is a study of molecular modeling from high throughput experiment, that is from the previous ones, and what we got it, the result is the large 10-fold selectivity. I'm going to address now another type of molecular interaction and how to design compounds. So in this, I would actually emphasize the tools used in chemoinformatics and QSAR. So here it will be more well-known chemicals, and it will be applied to tuberculosis. So we will design compounds starting from already known inhibitors and drugs against like tuberculosis disease, take the information out of it from chemistry information and attempt to design a new compound, and possibly we should find what it does target. So the next slide actually gives you an interesting picture. Now, please do note, if you have a picture like this is borrowed from a Chinese painter. He has painted most influential people in last 100 years, more than 100 years actually. And if I look at the picture or you look at the picture, it becomes very difficult to identify each one of them. However, if I know the person, how he will look at, or I will look for it, easy to find. Like I have shown here, Einstein was very easily found because his gesture or his shape, like having a long hair and mostly an old man. Similarly, we could very easily locate on the right hand side Mahatma Gandhi. He has a bald face and bald head, and then he has a very clean face with specs. We could identify him very easily. However, many other people would take much more time. I want to show you this picture to give you an idea. When you are doing research with a huge amount of data, you have to have some convincing concept for whom you are going for. So, knowing that is necessary. Next few slides, I will take you around to show you how we achieve from a huge amount of chemical C or chemical number of chemicals, how we identify and this kind of small some features and design the chemical afterwards. I would like now to address another interesting issue, which is if we do not have structures, if we do not have those information, but we have chemicals to design molecules, which is more like I would call chemoinformatics based research. So let us assume that there is a huge chemical space, which is shown like a chemical universe. Basically, one can have a rough hand calculations and can find almost it can be 10 to the power 40 to 10 to the power 120 compounds, all using only carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, and fluorine, chlorine, bromine, and iodine, as normally found in small inhibitor libraries, and the molecular weight should be less than 500. However, if we look at this structure space, we can as well find that there will be a biological space equivalent. That is not already known crystal structures in PDB. And if we have the structures, or we can use those structures to model our required structures, we can have a large biological space as well. So almost possibility of human genome is 30,000 or so, maybe much further larger for plants, and maybe much less available proteins which can be modeled and put it in our PDB space would be much less numbers for pathogens and other things. However, we must understand this space, this biological space is really expanding due to thanks to the newer methods coming up like electron microscope, NMR, etc. However, still we have this bottleneck of how I map the chemical space with biological space, which is shown here in this slide. We can select the biological space, but where is the ligand space? Which is cited by this final, on this same slide, by this call and very well-known phrase called drug research is search for a needle in a haystack. I would like you to show a case study where we would, if we know some kind of structure or needle, then it becomes much easier to find in the haystack where is the needle. So I would like to show you in few more slides and this case study. This addresses antimycobacterial chemicals, and we have taken from the references shown in the left hand side and build a large database which is now available even in NCI chemical databases and NIH depository. We have submitted to them, and here it shows that how we started working on it. So we first found the chemical structures, then what are the generic and their range of MICs. Why this range of MICs? Because they have been tested in a different methods. Number two, in different strains of mycobacteria. So we might have different these minimum maximums, and it's been shown in the range of MICs. Having this information, we can go ahead and develop a very good database structure, and then we start understanding from these chemicals. So this is a perfect example of data-driven research. So what did we do is we cluster them. Now you can make the molecules to be in fragmented. We can do fragmenting into global fragments, which is responsible for the activity of the compound across the database. And we can have local fragment, which will be active only in a same class of compounds. Keeping in mind these two clusters, we did a cluster analysis. However, on top of that cluster analysis, with chemical structure or fragments, we also did something called HQSR, which is a biological information added to cluster. I will go through the method to show you how we did it. However, the detail of the method has been spoken earlier module. So what we did is we found maximum common substructure amongst all these, which is shown in two slides here, which has been discussed earlier also. And once we get the subgraphs, then we started doing the global QSAR and local QSAR. This has been published papers also. And we portrayed on the right hand side every chemicals and their coloring of it. That means if it is the fragment which raises or provides favorable change, that is if it becomes a better binder or better inhibitor, then it's colored in a green, and whereas if it is not, it will be colored in a dark red. Once we do this clustering, we can identify few of these clusters, which is shown on the right hand side, as we have selected a cluster. Now this cluster are not chemicals, they are just a part of chemical. So fragmental, these clusters can be stitched together and make by using the right side picture like here, which shows you how this combination of structure can be shown. And that is, you have one scaffold yellow or scaffold 2, and this has been applied in very many other earlier example, where you can use the linked, which is something like connected, or you can have merged, which is shown by the next one slide, for either antagonist, two types of antagonist, or agonist and antagonist of different types. So this framework keeping in mind, we designed compounds from those fragments. I show you here. We did do the work after this, a QSAR. I show you standard traditional QSAR where the fragments would be taken and all the molecular properties can be calculated. Now in standard traditional QSAR, we cannot use fragments. Rather, we have to use the full structure activity. We develop the descriptors and we make the model with a formula like shown on the right hand side. What we did here was a different non-traditional QSAR, descriptors of fingerprints, holograms, etc. Now, what we did is, first we took the fingerprint, which says which that fingerprint is there or not. Then we divided into molecular fingerprint, and molecular fingerprint also not only gives you the numbers, and hologram fingerprint gives how many times it has occurred in that particular molecule. Using these techniques, we found out who are nearest and calculated the Tanimoto and Hamming distances, which shows you how the HQSR works, as these are holograms, are strip of numbers which have been taken to do the QSR. And we design the molecules. On the left hand side, there are the molecules, and here on the right hand side is a how we contribution. So the good contribution is shown by the shaded areas, that these are the portion which has contributed positively to make the inhibitor better off. Then what we did, whole summary. First, we collected an amount of almost 847 compounds. Now it has increased much larger in our database. Then we found the maximum common biologically contributors. Then we calculated. Now on the right hand side, you can see several different kind of targets might happen. Here we do not know even which will be the target, but we assume maybe one of these cubicles would be the target of that. So till now we don't know target, but we have designed a compound. Now we should test this compound. And what we did is shown synthesize this kind of motif scaffolds based designing at Naipar, Mohali. Well, whole pathway to find chemicals, from the chemicals, the scaffold I have described. But the question is how we have designed a scaffold which is shown in the center, but which target it should bind? This is another question. We must find that also, so that we can actually design the chemical better off, or we make a modification to increase its ability to bind the targets. That starts from how you trace back where from you got the scaffold. That means we should look into how we got the scaffold. So what we did was, to find this scaffold, we actually went to the database, which is shown in next. So we looked at the database, collected information as we started. We got the maximum common biologically active substructure, which is shown as small fragments. And then we designed the compound by doing the HQSR, earlier I have told you, and then got the hit list or design scaffolds. What we are going to do now is search our scaffold, how much similar the scaffold is in our main database. Then we identify, in this case actually we did identify that our scaffold has a very much resemblance with rifampicin. And we already know the rifampicin binds to the RNA polymerase. Hence, we selected next to search for targets using rifampicin binding RNA polymerase structure, and dock the scaffold there, try to optimize the structure and try to validate also whether it really binds and covers the interaction mapping, etc. or not. Actually shows where the in search of the target for our synthetic compounds. We already had in our databases rifampicin, which is very good inhibitor and known drug for tuberculosis. what we notice that our molecule, synthesized molecule, has a higher resemblance structurally with rifampicin. So we looked at the RNA polymerase structure, which is shown in this slide, elaborated its activity, etc. This is the crystal view for taking the rifampicin and binding at its active site, which is shown on the left hand side on the top. When we docked that same rifampicin, we recovered exactly the crystal structure and we identified which are the rifampicin interactive amino acid from the protein. Left hand side shows the blue module is the superimposed or overlap of our compound, synthetic compound with rifampicin. As you see, rifampicin is a large compound, large molecule, and actually it is a product from one of the natural fungal system. However, ours is a designed compound. So what we did is we used the same motif, we tried to dock in the same place as in rifampicin, and we did this rifampicin binding to with a tuberculosis moiety or RNA polymerase of tuberculosis. We actually molecular modeled that and then docked it. And next slide shows the values, etc. What we got the docking systems. How we docked the molecule. So this is left hand side is our compound. We can have many combinatorial. Now, compound was docked inside active site, which is shown by dash, and the color code of this molecule, which is the amino acid, interactive amino acid with our molecule in the RNA polymerase active site. This is the proof that our model or our hypothesis was right. The reason is we have synthesized many compounds, out of them 4 compounds, which is shown on the left hand side, the numbers, and their heats. This is the compound moiety and heats under heats. When we did the experiment, accomplished was with tuberculosis inhibition at constant microgram to 2.5 microgram per ml, and we did with Bowie's experiment with Bowie's, which is also similar at MIC calculations. We figure out the best compound shown is C1. That means it is having 98% inhibition for H37RV, which is a pathogenic mycobacterium. In addition, it inhibits almost 90% at very low 2 microgram per ml. And this data shows also that SAR is accomplished because consistently you see by changing 1 or 2 fragments, we get a very good consistent values of increment or decrement with 90% inhibition of M. bovis. Using these molecules, we docked all of them and we took for the next study. How we have posed with respect to bioactive RIF. RIF was always used as a standard for our activity or putting it in pose, recovering the pose, and we got the best inhibitor, which is C1, with a very gold score was -14, whereas this is -140. Higher, the lower the score is, worst is the values. So we have calculated these 2 scores and found out which will be the most effective. It shows actual interaction of each one of them. Now, as you see on the gold score, that the compound, which is C1, which has the best activity, earlier also we have shown, has the -1389, that is the lowest minus or highest minus gold score. And that the best part is to look at not only the value of scoring, we rely more on flexible docking. And what we figure out once we did the good flexible docking, this many interactions are satisfied. Not only that, we put another molecule which has a very similar structure called plumbagene. Literature showed that it also have very good inhibition quality. So we took that as well to have our data to be validated, and we figure out plumbagene also has a very nice nearby value from as much as ours. Of course, our compound scores much better and does much more interactions with the active site, which is satisfied also by rifampicin. In summary, so I have shown you one is structure-based drug designing procedure where we have actually identified by high throughput, some experimental high throughput, some of the chemical motifs or chemicals, and designed and modified that in active site to find a selectivity against or favor of malaria and against the human host. I also showed you database driven, that is chemical database driven anti-tuberculosis compound modification, and I have shown you how you can have the different types of problems to solve, how you can use different types of bioinformatics and chemoinformatics tools. So what could see here that as long we are having protein structures, etc., and we can actually identify chemicals, we can find a proper docking and interaction mapping and calculate even interaction energies and design compounds. We can do a selectivity, as I have shown you in case of antimalarial. We can as well do design a novel compound which will compete for some new compounds or new targets if possibly. However, I have not talked about how the case can be more complicated when the protein or the target we have to make it to design is very flexible, which I will follow in different module. Thank you.